Yay! Protein synthesis. In this module, you are going to learn how to transcribe and translate a gene in order to synthesize a protein. So, genes. What do we know about them so far? You know that you inherit your genes for things like eye color and other important traits from your parents. You inherit a bunch of them from your mother and a bunch of them from your father. Maybe your mom gives you a big B for brown eyes, and your daddy gives you a little b for blue eyes, and therefore you're a heterozygote, and you'll end up with a dominant allele, brown eyes. So, how do we go from genetic information to brown eyes? Well, let's talk about your eyes. Your eyes are composed of a bunch of cells, and all these cells are very specialized. These cells in the iris of your eye, right here, make a protein. And that protein just so happens to, let's say, reflect blue light. So when blue light is reflected off of those proteins, people see your eyes and they say blue light. And so they see that your eyes are blue. So these genes you inherit from your parents are used to make proteins, and these proteins give you the traits you have, and they keep you alive. These are the machines that do the jobs of life. So another example of a protein is the protein hemoglobin, right? And this is a protein that is found in your red blood cells, and its job is to carry oxygen from the cells in your lungs to all the other cells in your body so that you can break down your food to make ATP. So protein synthesis. We're going to go from a gene to a protein. So that is the point of this exercise. How can you transcribe and translate a gene in order to synthesize a protein? Well, the first thing you need to know is that it's a two-step process. In the first step, the genetic information in your genes, which are found on your DNA, are used to synthesize a messenger RNA molecule. You will recall that this messenger RNA molecule is a message sent from your DNA to a ribosome whose job it is to make that protein. That's the second step. So the first step here, where we're using the information to make an mRNA, that's called transcription. Transcription. Transcription happens in the nucleus of your cells. Inside of the nucleus of your cells, that mRNA is going to be synthesized. That mRNA, after it is transcribed, is going to leave the nucleus through a nuclear pore or a hole in the nuclear membrane, and it's going to go out into the cytoplasm where it will find a ribosome. Step 2 of this process, which is called translation, is when the ribosome takes that mRNA and it uses the information in that mRNA to assemble a bunch of amino acids into a protein. Now, what amino acids go into that protein? Well, that depends on the protein, right? So the amino acids that make up the protein hemoglobin, that's a different sequence of amino acids than the protein that makes your eyes blue. So let's look at this process in detail. Step 1, or transcription. To make a protein, a cell first transcribes a segment of your DNA, known as a gene, into an RNA. So over here, I've got a model of this process, and I want you to follow along with me and take careful notes. The base pairs of DNA are first unzipped. And they are unzipped so that the cell can expose the gene. The gene is that segment of the DNA which contains the information to make a protein. So here in our model, you see that the DNA has come unzipped from there to there. One of those two sides, here the yellow one, actually encodes the genetic information. It is called the gene. The other strand is called the non-template strand. So that's the non-template strand, but the strand that includes the information or the gene is also referred to as the template strand. So the base pairs of DNA are unzipped, and a special protein whose name is RNA polymerase actually transcribes one side of the ladder to make a single-stranded messenger RNA molecule, or an mRNA. In the figure, you see the gene here, which includes a sequence of nitrogenous bases, A, T, G, C, so on and so forth, all the way to the end. And then down here, you see the mRNA being assembled. This mRNA is being assembled by a protein called RNA polymerase. Now, as RNA polymerase makes its way down the gene, it's going to use the bases in that template to create this mRNA. So when that RNA polymerase senses the A, it will place a U in the RNA. Remember, RNA doesn't have any T's. It uses U instead. When RNA polymerase sees a T, it places an A in the mRNA. Then it sees a G and adds a C to a G, and so on and so forth. But recall that the A's give rise to U's on the other side, not T's. So we'll go ahead and finish our mRNA here. Okay? And so then we have an mRNA molecule. That mRNA molecule is then going to leave the nucleus. Step 2, translation. After the DNA has been transcribed into an mRNA molecule, the mRNA molecule is then translated into a protein. Translation occurs in the ribosomes of cells. So in my figure down here below, I have got a chromosome here in my bacterial cell that is a single circular chromosome. So it's a little bit different from your chromosomes because your chromosomes are inside of a nucleus. Bacteria don't have nuclei. They are prokaryotic. Also, their chromosome is circular, whereas yours are linear. Other than that, they're pretty much the same thing. So up here at the top, what you see happening is RNA polymerase is transcribing an mRNA. The mRNA is represented by that pink ribbon that's being synthesized by RNA polymerase here. You see now that this mRNA has found a ribosome, and that ribosome is now translating the mRNA to make a protein or a polypeptide. You will recall that the amino acids in a protein are held together by these special bonds that we call peptide bonds. So, a polypeptide is just another word for protein. So, that's protein synthesis in a nutshell. So now let's practice. Let's start with our gene. So here's my DNA sequence in my gene. T, A, C, G, 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 A, T, T. Now in real life, genes are anywhere from hundreds of bases in length to tens of thousands or even hundreds of thousands of bases in length. We don't have that kind of time. So our gene has 9 bases. So step 1, we are going to transcribe the DNA to make an mRNA. So, you ready? Let's do that. Okay, so we are going to take on the role of RNA polymerase. We see a T, so an A must go there. All right, moving on down, we see an A, so a U must go there. Remember, mRNA does not use thymine. Instead, it uses uracil. The C will give rise to a G there, the G to a C, the G to a C, and the G to a C, and the A to a U. Ha! The T to an A, and the final T to an A. Ladies and gentlemen, you have created your first mRNA. The next step, step 2, is translation. So this mRNA is now going to leave the nucleus, and it's going to go find a ribosome. That ribosome is going to take that mRNA, and it's going to use the information in that mRNA to make your protein. Now, a protein is a sequence of amino acids. So how, might you ask, does the ribosome take this information that you've made into an mRNA and turned it into a protein? Well, we're going to use the genetic code. You've heard of the genetic code? Here it is. This is the key by which your cells, your ribosomes, take the information in an mRNA and make a protein. This code is written in 3-letter words. So the first thing I'm going to do is I'm going to count my bases in my RNA. 1, 2, 3. And after those 3, I'm going to draw a little line. And that line's just there to keep me organized. Again, we're looking at 3-letter words. So the next 3 bases, 1, 2, 3. Now I'm going to put another line to keep myself organized. And then I'd continue with the next 3 bases, 1, 2, 3. This just so happens to be the end, but I'm going to go ahead and draw my line anyway. All right, so what we've got here are 1, 2, 3 3-letter words, and these 3-letter words are called a codon. A codon is 3 bases in an mRNA that code for an amino acid. So, since we have 3 codons, what we're going to end up with is a protein with as many as 3 amino acids. So this is our amino acid number 1. This will be our amino acid number 2, and so on and so forth. Okay? So, let's look at our first word. Our first three-letter word is A-U-G. First base A, second base U, third base G. So now let's pretend to be ribosomes, and let's translate this AUG into an amino acid. So again, that codon was AUG. Here's how you use the genetic code to figure out what amino acid AUG stands for. I want you to notice that over here on the left, it says first base in the codon. Our first base in our first codon is an A. So here's the A right there. That means that our first amino acid must be one of these 16. Isoleucine, methionine, and the others. Serine, arginine, lysine, so on and so forth. Our second base in our codon is U. You'll notice at the top it says second base in the codon, so you're going to find the U. Where's that U? There it is right there. So now I know that my amino acid has got to be somewhere in this column. So I'm going to go down to where it overlaps with my first base, and there it is right there. So now I know that my amino acid must be one of these four options: isoleucine, 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 or methionine. So which one? Well, I got to look at my third base in my codon. My third base in my codon happens to be G. So I'm going to look for the G in this segment, and there it is right there. So I'm going to follow that G all the way over to my little intersection. There we go. We found our first amino acid. Our first amino acid is methionine. M-E-T. So our first amino acid is M-E-T, or methionine. You can just write M-E-T. My next codon number 2, is C-C-C. So let's translate C-C-C. So if my codon is C-C-C and my first base is C, well, let's find the C. Okay, so I know it's one of these 16. My second codon is C, so it must be one of these 4. Well, I guess it's got to be PRO. But just to make sure, let's look at our third one. And our third base is C. So I'm going to follow that C over, and yep, it's proline. So my second amino acid is proline. Now let's read our third three-letter word, our third codon, UAA. Well, let's check this out. So we've got our first 2 codon thread. Let's see what our last one is. Again, the codon is UAA, UAA. My first base in my last codon is U. So let's see, there's U. So I know it's going to be one of the ones up here. And my second base is A. So I know it's got to be one of these 4 options here. And my third base is A. So I'm going to look here, and oh, there's A. I'm going to follow it over, and look at that. It says stop. I wonder what the ribosome should do here. Let's see. Well, I guess it should stop, right? So what you've seen here is a stop codon, and this stop codon tells the ribosome to, tells it to stop. So you're done. So the question is, how many amino acids are in this protein, and what are they? There are 2 amino acids in this protein, methionine and proline. Stop is not an amino acid. It is an instruction to tell the ribosome to stop. So this protein right here, that protein It's going to go now and do its job. If its job is to carry oxygen to all the cells in your body, that's what it's going to do. If its job is to absorb and reflect light, then it might just be absorbing and reflecting the light that makes your eyes blue. Ladies and gentlemen, congratulations. You can transcribe and translate a gene to synthesize a protein.